Hello TY BSC. In last lecture we have started with the Ascaris lumbricoudes. We have seen the habit and habitats of Ascaris lumbricoudes. Then we have gone through the morphological characteristics of Ascaris. In that, as we know that sexes are separated in Ascaris lumbricoudes. So as you can see, we have differentiated between the male and female. In that we have seen male is smaller than the female. In general characteristics, we have seen both have the tapering and mouth parts, excretory parts are present. Along with that, males have spicules towards its anus, and females have female gonopore as well as the lateral line, and other than that the papillae is also present. So these were the morphological characteristics that we have seen in previous lecture. So today we will start with the reproductive system, the life cycle of this parasite. So in first of all, in the reproductive system, it is made up of single long genital tube with number of coils in the posterior half of the body cavity. See, while reading this part, reproductive system, always keep the diagram in front of you so that you will come to know what exactly we are talking about and what it is. So if you talk about a single long thread like coiled testis joined to vasa deferens of the same diameter, then just look at this diagram. Here you have the testis, then you have the vasa deferens. So this testis is joined to the vasa deferens and the diameter of this tube is similar. Vasa deferens then further joins to a broader seminal vesicle. So as you can see, the diameter of this tube increases. This makes the difference that this particular part is vasa deferens, and after this you have the seminal vesicle, which is somewhat broader as compared to the vasa deferens. Now this particular seminal vesicle leads into the narrow tube. Now here you have a narrow, then you have a broad, then again you have the narrow tube. So narrow muscular ejaculator duct that opens into the cloaca at this position. So this particular tube, it becomes narrow. This is a ejaculator and it opens into the cloaca, and the pouch contains spicules. At this particular area there is a spicules that is useful in copulation or copulatory. It is one of the copulatory organ to open the female genital pore or the vulva. Okay, so testis is telogenic, that is the terminal portion contain mitotically dividing spermatogonia. So where exactly this mitotically dividing spermatogonia occurs? Present at this position. From here, this whole position you have testes, then from here the vasa deferens, it travels to the vasa deferens, then it travels to the seminal vesicle, and then at the end cloaca. So this is all the reproductive system, male reproductive system. Now we will talk about the female reproductive system. The female reproductive organs are diadelphic, means double. Remember this word, diadelphic, and occupy posterior 2/3 part of the body. Posterior 2/3 part of the body is occupied by this particular reproductive system. What it consists of? It consists of ovaries, oviduct, uteri, and vagina. Okay, so consists of ovaries. Where are the ovaries? Look at this in this diagram. You have these ovaries, then you have oviduct. Ovaries are there, then you have these oviduct. Okay, from here onwards the whole thing is oviduct at this part up to this part. Okay, then you have uteri and vagina. This is the vulva and this is the vagina. Okay, so ovary from here up to this part, then you have oviduct from here up to this part. This is the area where uterus containing the eggs. Then you have the vulva and the vagina. So two long, highly twisted threads like ovaries are the tubes joined to broader oviduct. So this is the long tubes. Each oviduct further continue into still broader, thicker and muscular uterus. From here onwards, as you can see, the uterus go on increasing, it become broader and broader, and at the end you have a broad vagina and vulva also. Vulva is not broad, but vagina, up to vagina. Okay, so the first part of uterus functions as seminal receptor for sperm storage, where fertilization takes place. Remember, so first part of uterus, okay, that is responsible for sperm storage, where fertilization occurs. The remaining uterus stored fertilized eggs. So remaining uterus stored fertilized eggs. So vagina is formed by union of two uteri and opened by transferred gonophore. So this vagina actually formed because of the union of two uteri. The tip of the ovary produce oogonia. Generally, how it is produced? So tip produces the oogonia, and then oocytes maturation occurs in uterus. So oocyte maturation occurs in the uterus. So the interaction between the sperms and the oocyte cells occur in seminal receptacle. Now, Ascaris male and female have unequal number of chromosomes. Remember, this is very important. A question can be asked in some competitive exams also. So they have unequal number of chromosomes. Both are not having equal number of chromosomes. If we talk about humans, then you have a 23 and 23, male and female. Okay, in any other animal also the equal number of chromosomes are there in male and female gametes. But here in case of male and female gametes of Ascaris, male as having 43 chromosomes, whereas female having 48 chromosomes. So there is a difference between these two. Now we talk about the life cycle, how the copulation and fertilization takes place, and after fertilization what are the stages of development. So mating or copulation takes place in the host intestine. Remember, they grow in the host intestine. Okay, so the last stage or adult stage, they will come together in the host intestine. During copulation, male Ascaris move in such a way that the cloacal aperture, okay, the cloacal aperture faces the vulva of the female. Remember, in previous diagram we have seen, from the cloacal aperture you have the tube which contains the sperm, and from the cloacal aperture it will come out, and nearby the cloacal aperture itself, see, as you can see, the ejaculatory duct, and this is the cloacal aperture. Okay, this is the ejaculatory duct. So this cloacal aperture comes in front of vulva of the female. Okay, now what happens then? Male project out these copulatory spicules to open the vulva of female, and then the cloacal wall of the male contracts, causing transfer of sperms into the vagina of the female. Okay, so this particular part, it opens up the vulva, and the ejaculatory duct where you have all the sperms present, it is transferred into the vaginal region as shown in the diagram. Now the cloacal wall of male contracts, causing transfer. The sperms come to lie in the seminal receptacle part of the uteri, and then wait for the ova to come through the oviduct for the fertilization. So number of sperms will be present in this area, and it will just wait. So the eggs or ova will come, and the fertilization should takes place. Okay, so just look at the diagram in magnified form also. So these are the important parts: ejaculatory duct, this is the rectum, cloaca, and then you have a copulatory spicules, and this is the vulva, and this is the vaginal area, this is the vagina uterine branch. Now once the fertilization takes place, then what happens? So occurs in the seminal receptacle, just now I have told you, entire sperm enters the ovum and followed by completion of meiotic division of oocyte and chromosomal pairing. Now after fertilization, immediately the glycogen globules of the egg migrate to the surface to form the fertilization membrane. Now initially, as you can see here in the diagram, this is the fertilized egg and this one is the unfertilized egg. This is the difference between two. So before unfertilized, it has amorphous material. After fertilization, as you can see, there is a lot of change that has happened here. Both are having the albuminous layer, white line layer is present in the fertilized egg, chitinous layer formation is there, lipodial layer formation and vitaline envelope formation also takes place in the fertilized egg. So as you can see, the hardening of this chitinous layer takes place. Fat globules of the egg form the lipoid layer below the chitinous shell. This is the lipoid layer, as you can see. Okay, and the fertilized egg passes down, during which the uterine walls secrete an outer thick yellow or brown albuminous coat around it. So this is the albuminous coat. So what development takes place after the fertilization? Development of the chitinous layer, lipoid layer, lipoidal layer, vitaline layer and albuminous layer, remember. Okay, so there is one characteristic wavy surface of this particular albuminous layer. Okay, and also such eggs are sometimes called as mammaliated egg, remember. So now after this, the explanation about the egg formation. So eggs are elliptical in shape, measure about 60 to 70 micron. Okay, so this is the fertilized egg that has been formed, and female Ascaris produces many unfertilized eggs which may have more elongated capsule. Its shell is also thinner without vitaline envelope, remember, which is not fertilized, it will not have the vitaline envelope. Okay, then you have the amorphous refractive matter. Both uteri may contain about 27 crore egg, and everyday female can lay about as you can see 2000 eggs. So a lot of number, but as we know that such kind of adaptation has been observed in many parasites. Why there is a lot more production of egg? The reason is lot of egg is lost and it may not find another host. That's why, to increase the chances of infecting other hosts, the number has been increased. What is the lifespan of female in 10 to 12 months? So mature female can produce more than at least 6 crores of eggs in her reproductive life. So 9 to 12 months the female may remain within the intestine, and during that process it will continuously produce the eggs. Large number of fertile eggs also show rich mortality rate. Okay, so very few infective larvae reach the human intestine. That's why I have told you that that's why they produces such a large number of eggs, because all doesn't get the host, all will not get the chance to infect the human intestine. So one more thing, why the ideal environment for completion of life cycle and continuation of species is outside of this human body. So these eggs, they come out. Okay, we will see this diagram again. So how the development takes place, that is important. So in the development, here from the life cycle begins. Fertilized egg stage development shows metamorphosis including series of 4 larval stages to the adult form. Remember, before coming out of the egg, first of all you have the development of the egg itself, the zygote formation, then you have blastomere formation, then you have morula, and so further more development will takes place, and at the end you will have the larva present within the egg. So the development of egg and first 3 larval stages occur outside the human host in the soil. Remember, it cannot develop within the body of humans, it has to go outside of the body, so in the soil where eggs are deposited. So infective larval stage when enters the gut of human host shown parasite life and develop into the host. So this is the diagram. As you can see, different stages. The first stage is zygote, then you have blastomere formation, then you have morula, and further development takes place, and at the end you have the first stage larvae which is present. So what happens with the egg when they are deposited into the soil? So eggs survive in the soil, but the only problem is the soil or the condition should be moist enough. If it is highly dry, then they cannot survive. But as we know, egg capsule wall is permeable to water and gases, but highly resistant to cold and warm temperature, strong chemical and desiccation. Also it has survived the 10% formalin solution and the sea water also. It cannot survive the high temperature, that is 70 degrees Celsius. Also it may alive in the ground for longer duration, but as we know, if it is highly drying condition, then it may not survive. Within few days it will die, nothing will happen, and or otherwise it will remain there for months. So development of egg require oxygen, moisture, shade and temperature. Remember one thing, that the development of egg require oxygen, and human intestine there is a deprived of or lack of oxygen. That's why this development require oxygen, so they should come out of the body, they should not remain within the body. Okay, and temperature lower than the human body, this is also important for their development. The temperature should be lower than human body, that is, it should be lower than 37 degrees Celsius. Most favorable is 34. Okay, so it cannot survive within the humans, it has to come out. Okay, now in the favorable condition, larva, first stage larva formation may takes place. So one by one, first cleavage to blastomere formation is there, forms ectoderm, and another give rise to endoderm and mesoderm. Then blastomeres form blastula at 16 cell stage having blastocele. Then the gastrula is formed by epibole or overgrowth of ectodermal cells, and then by the end of 10 to 14 days worm like juvenile is formed. So these are the stages. Okay, now this worm like juvenile formed, as you can see within the egg, it possesses the alimentary canal, a nerve ring and a lateral excretory system. Remember, also the juvenile is elongated worm like structure with rounded anterior end and tapering body. Remember, it's a juvenile. What we have seen in the morphology of larva of the adult, both are having, male or female, both are having tapering end at both sides. But in case of juvenile, you have the rounded anterior end and tapering tail. So one side is rounded, it's not tapering, where another side is tapering, and there is an envelope of very smooth cuticle. So large number of granules are present in the body which are absorbed or used up after first mold. Okay, now the juveniles are very much similar with Rabbitidis nematodes, that's why they are sometimes also called as Rabbitidis or Rabbitoid. So this is infective stage to man before hatching. Larva undergoes the molting. This is the second stage, as you can see, excretory cells. So it is formed inside the egg capsule. Okay, first instar larvae molt in 7 days and becomes second stage juvenile. As you can see this diagram, this is the second stage juvenile. Okay, larvae show digestive, excretory and nervous system as well as sense organs. Excretory pore, and here as you can see there is one tapering end and this is rounded end. Both sides are not tapering. And the nerve ring, here in this diagram you can see the nerve ring. This is the third stage larvae. Okay, it developed after 10 to 14 days. Environmental temperature plays an important role for this. Whereas here excretory cells, genital primordium is present, and by the end of 7 days the larva become 2 to 50 micrometer long and 14 micrometer in diameter, molds into next instar larvae. So this is the difference between the two second stage larvae. This is the third stage larvae. So this stage is also infective to man. If this larva does not get entry in the body of host, then it can remain viable under favorable condition of oxygen, moisture and temperature for about 6 years in the soil. Remember, it may remain, but yes, moisture is required. Anything, if it become dry, then it may not survive. Okay, so as you can see in this diagram, there are different stages. First thing, you have the hatching larva, then you have the first second stage larva, then you have the third stage larva, and this is the fourth stage or adult larva, where you get the esophagus, the developmental gonad, intestine is there, and now ring is also there. Now, how the infection to man takes place. So there is no secondary host. Remember, here only one host is there. He is the definitive host, that is man. So no secondary hosts are there in the life cycle of Ascaris. So eggs go out, development takes place, eggs should again enter into the body of humans for this purpose. So how this thing takes place, okay, that we will see. Infection may occur through the contaminated raw vegetables or food or water with the infectious, where the infectious eggs are present, or we haven't washed it properly and then using it, then you may get the contamination. Also through the soil contaminated fingers. We know that small children, they always like to play in the soil, and within the soil you may have these eggs. So ingestion of these eggs may take place through the dust also. Now embryonated eggs pass down the small intestine first of all, as these are covered or protected. That's why what happens here, the egg, whatever egg you have, this embryonated egg, it passes through the stomach to the small intestine, the duodenum. Here egg capsule become weak due to action of carbon dioxide and pH and host digestive enzymes. Larva inside the eggshell also get activated. So larva secrete a hatching fluid inside the egg capsule containing enzyme like esterase and chitinase. Remember, because chitin, that egg is having just now I told you about the layer of fat and the layer of chitin. Now, a layer of fat is digested by esterase, whereas layer of chitin is digested by chitinase. So these enzymes make shell very weak and alter the structure, and larva form comes out of it. So process called as hatching. This process is called as hatching, and it takes about 3 to 4 hours. So the juvenile are about 0.2 to 0.3 mm long, 13 to 15 micrometer in the diameter. Now they all have organs in adult except the reproductive system. Development in the host, after reaching the definitive host, the larvae do not remain in the intestine for the further development. but go to the typical wandering tour of 10 days in the host body. So it will not remain as it is. So what happens then? Newly hatched larvae bore through the intestinal wall and carried along the portal vessels to the liver, where they live for a period of 3 to 4 days. Now here the transfer of these larvae takes place. Okay, so a lot of detailing are there. So I'll just tell you about the details how it takes place, okay, in the next lecture, because once I start this, then there will be continuation, because the entry point, then where they travel and how they get entry into the stomach, this whole thing is very much important. So this part I will explain in the next lecture. That will be the part 2 of this particular lecture. Thank you.